Sahli's method. Now, whenever you are from the exam point of view, just don't talk. No, don't disturb others in the class. Don't make sounds. SAHLI Sahli's method. Those of you who were there on that 23rd, that Monday already know. So, but the method name is Sahli's method. Now, from the exam point of view, when you are asked what is the name of the method by which you are estimating hemoglobin, but the examiner will come after you have done the work and will say what is your experiment. Okay, estimation of hemoglobin. What have you done? So, what is the name of the method? So, why I have given a gap between Sahli's and method? Because if you tell Sahli's method, the answer is partially correct. So, you have to tell. Sorry, that is very hazy. I'll repeat it for you. Sahli's acid hematin method, this should be your answer. Because estimation is being done on the principle includes the formation of acid hematin and then we estimate hemoglobin. So, formation of acid hematin, because we are using an acid which is, in this case it is hydrochloric acid, and reaction of acid with hemoglobin forms the compound acid hematin, and the complete answer is Sahli's acid hematin method. You may find in the books or wherever, or just when we, the faculties, the teachers are speaking with you, they simply say Sahli's method. We simply say Sahli's method, but the correct, not the partial answer, full answer is Sahli's acid hematin methods. As in Leishman stain, along with eosine and methylene blue, you have to also tell it also contains methyl alcohol and it should be acetone free. That is eosinated methylene blue in acetone free methyl alcohol. This is the complete answer. Here the complete answer is Sahli's acid hematin method. Now what do you do? This is the total apparatus for estimating hemoglobin is called hemometer. It is written over here, hemometer. Meter means measuring something. Hemo related to blood. Hemoglobin, here hemo means related to hemoglobin. So, estimating hemoglobin by the hemometer, HEMO, METER, some people write it as HEMO, that is the difference between English and American spellings. Here, obviously, hemometer. So, hemoglobin also HEMO you can write, or HAEMO. So, hemometer, it consists of the following things. This is something called the comparator box. The box like structure is called the comparator box. We are coming to its use. Those who have done, they already know. So, I think this is distilled water. So, distilled water will be supplied to you, or maybe distilled water, or maybe buffered water, distilled water. So, actually the apparatus consists of, main parts are comparator box. This is the water, leave it. This is the hemoglobin tube used for the estimation of hemoglobin. This is the stirrer, steering the fluid in the tube. Sorry, this is not the stirrer. I'm sorry. This is the hemoglobin pipette. What's the difference between a tube and a pipette? It was just my slip of the tongue, nothing else. This is the hemoglobin tube, this is the hemoglobin pipette. Now, what is the difference between tube and pipette? You should already know after, as total count has been done. In others, the past years we did hemoglobin before total count, but this time total count of RBC and hemoglobin has been done. So, just tell me quickly what is the difference between a tube and a pipette. This is the pipette and this is the tube. Now, what is the difference? The structural difference you can all note from just by looking at it, even from far off. What is the structural difference? The basic structural difference. What do you note? Capillary, the word capillary is not important. Simply what we can notice, a pipette is open at both ends and the tube is closed at one end and nothing else. Tube is closed at one end, pipette is open at both ends. Okay. So, tube you should put some fluid or whatever you are taking from one side, mainly obviously fluid, some fluid, and it will not go out from the other end, and you don't, and tube pipette we usually suck some fluid with the help of a pipette in the lab. So, as in case of RBC pipette, WBC pipette, there are no tubes in that, but hemoglobinometer or hemometer consists of both. It can also be called hemoglobinometer, Sahli's hemoglobinometer. It consists of both the pipette and a tube, both one pipette and a tube. So the difference with the RBC pipette, this pipette, the difference is that there are so many markings over there. Here there is only single marking in the form of 20, and this 20, another difference is, so one difference is it has a single marking is single. Another difference is there the markings were, in the case of RBC and WBC pipette, the markings are 0.51 and 101 in case of RBC pipette, 0.51 and 11 in case of WBC pipette, but without any units. There are no units that just indicate parts of volume. Here this 20, it is also written, the unit is written over here. Here it contains a unit, it is microliter, and this much, taking this much blood means taking 20 microliter of blood, and we do it taking by exactly more or less, if not exactly more or less 20 only. If it should be up to this mark, so it is exactly 20 microliter of blood. And there we could, even in the case of RBC and WBC, instead of 0.5, if we take blood up to 1, obviously dilution will be less, but still the experiment is theoretically possible. Here whatever we do it with 20 microliter, not half of it, not double of it, not 40, not 30, not 10. We do it with 20 microliter because the experiment is based on the principle where it has been standardized that we will use 20 microliter of fluid, and acid we will take before, after drawing the blood in the pipette 20 microliter. Before that, not after, sorry, before that we already take acid here up to, there is a mark yellow. You do not follow red or yellow, but what the meaning of yellow is yellow mark 2. That means this is in gram percent. Hemoglobin, we measure hemoglobin, theoretically we know hemoglobin is 14 to 16 gram percent or 12 to 14 gram percent in case of females. So 14 to 16, this gram percentage is given in, this graduation is given in yellow, and some reddish graduations are there, red graduations, which we do not use. That is just the percentage of hemoglobin. Here it is the percentage of hemoglobin, the red ones are 20, 40, 60, 80, 100 like this. 100% corresponds to 15 gram percent. The graduations are given like that, but that is just for the manufacturer is still manufacturing like this. We do not need that. The method we use, we do not need the percentage graduation. You can just compare that 100% equivalent to 15, but graduations, we need the YOLO graduations. You won't tell YOLO, you will tell this is the gram percentage graduation, gram percentage or gram per DL, gram per DL of blood, deciliter of blood. Deciliter of blood is also called gram percent because 1 deciliter actually is 100 milliliter. So gram per 100 milliliter is gram percent. So hemoglobin is expressed in gram percent. We take acid up to 2 gram percent. The 2 gram percent mark, we put the tube in the box. The comparator box has one, see the comparator box has 2 fixed, not tubes, 2 fixed, this fluid like this colored colors. Comparator, this is one of the colorimetric methods. That's color will be compared. 2 colors, similar colors are there on the 2 sides, and in the middle we can put, there is a this place for placing this tube almost just greater than the diameter of this hemoglobin tube, where we can put this hemoglobin tube. So after taking acid into up to the 2 gram percent mark, there is no 2% mark, it starts from 20%. That is only percentage which we do not need. 2 gram percent, which is yellowish over, usually yellowish, but you will go by 2, 4, 6, this is level, this graduation, even if it is some other color. So the color is not important, 2 gram percent mark. Taking acid up to the 2 gram percent mark, we put it in this hemoglobin comparator box, and then we suck 20 microliter blood, 20 up to the mark of 20 microliter, and as soon as we suck it, we just then transfer it to the tube where there is the acid, and just by very gently mix it for say maybe 15 seconds, and then add distilled water. 10 seconds, 5 seconds, 10 seconds, 15 seconds, add distilled water. Now distilled water should be added slowly. Distilled water will be added with the help of a dropper which is given over here. help of a dropper and putting 1 or 2 drops of distilled water. This is the stirrer. This glass rod is the stirrer. Just stir the fluid, then put the fluid, then again put the tube here, and we go on comparing. So we have to, bright light should be used. The color of the acid and the blood mixed with it will be very dark, much darker compared to what are the colors given on the two sides. Now it will gradually fade. Fade means it will gradually, by dilution with distilled water, the color will become lighter. And we go on mixing distilled water, stirring it till we get the color of, it matches the color which is in the comparator box, which is already the standard color given in the comparator box. Now for the beginners, for the first day, when you are doing on the first day, it is okay that you just match the color. When the color gets matched, whatever is the reading, whatever is the that means the level of the fluid in this hemoglobin tube, if it is at say 16, say 14, then the value of hemoglobin estimated is 14 gram%. But ideally it should be 3 readings, just deeper than the standard color, just deeper than it is going on diluting. It was very dark color, not deeper, just darker. It was very dark color, now it is becoming lighter. Now as it is just darker, you can just, by your sense, if you, whatever you are sensing, that after, if I add distilled water after this, it will match the color. So just darker, you take one reading, maybe it is at 12.8. Between 12 and 14 there are more graduations, there are smaller graduations. Just say at 12.8 it is just darker, then you give another drop of distilled water, it just matches, say it is at 13 or 13.2. So that is one, you take that reading and then you add another drop and it becomes 1 or more, 2 drops more, it becomes just lighter. So you take that reading also, you take 3 readings and take the average. This is what should be ideally done, not the single reading, because it actually depends, it is human eye variation. So somebody will tell, no, at 12.8 I think the color has matched, somebody will tell at 13.4 now the color has matched, it did not match in between. So better you take 3 readings when you are taking darker, when you are feeling, say you are feeling it is just darker but your friend is feeling it is just, it has already matched. So you take 3 readings, okay, it is just darker, I take my reading, you tell your reading, and just lighter, you add 1 or 2 more drops. But these readings will obviously not be very far apart. Even it won't be 1 is 12, 1 is 13, 1 is 14. It may be 1 is 12.6, another is 13, another is 13.2, or 1 is 12.8, another 1 is 11.8, 1 is 12.2, another is 12.4 or 12.6. But it will not be like that 1 is 10, 1 is 12, 1 is 15. There will be not so much variation. It will be in one decimal may vary, but not the whole number. So that much variation should not be there. It is just for eye estimation, human to human difference. That difference, so 3 readings should be taken and 1 reading will. So this is a very simple thing to do. Only simpler thing to do is, I think, blood grouping and BTCT, but compared to total counts and differential counts, this is a very simple experiment. And obviously after the experiment is done, you will face questions when you do in the exam. So doing the experiment correctly and answering the questions correctly, there is 10 marks. Although if hemoglobin, as it is a simple experiment, along with hemoglobin you will get something, either bleeding time or clotting time. And also you remember, in the exams questions will not be direct questions like estimate hemoglobin of the given blood sample, or sometimes maybe you prick your own finger, nothing, you prick your own finger, estimate hemoglobin. Usually questions will not be that type. So after total count of RBC and hemoglobin is done, you will face questions on RBC indices. So mean corpuscular hemoglobin, if it is one of the RBC indices already discussed in the theoretical classes, one is mean corpuscular hemoglobin. So mean corpuscular hemoglobin is, you require both the hemoglobin count and the RBC count. Any one will be given in the question, usually where you won't have to perform both. Any one will be given in the question, another you have to estimate, and then you have to calculate MCH. That is your work in the lab, and that is in 45 minutes. But as hemoglobin is a short experiment, along with hemoglobin you will face something like bleeding time or blood grouping or something. If it is DLC or total count, then you will face one experiment. Usually in the exam it goes like this. This is from the exam point of view. In 45 minutes, either differential leukocyte count or total count of RBC or total count of WBC. Again, total count of RBC may not be a direct question. Say MCH in RBC index, you have to calculate. Hemoglobin percentage is given in the question, you have to calculate MCH. So you should have to estimate the total count of RBC, but there you will face one experiment. Okay, but if you are to face hemoglobin, then you will face either bleeding time or clotting time or blood grouping, some other short experiment along with it, and 2 experiments you have to complete in 45 minutes. So in the class also you go like that. Today you will get about, say, what is the time? Has my clock stopped? Okay, it's 11:20. Right. So today, say 11:45 or 12 noon to 1 p.m. you should do both. 11:40, 1 hour to 1 hour 15 minutes you have to today do both. That means you have to, for most people hemoglobin is the only class. So you have to do both. So hemoglobin, estimation of hemoglobin you do. So this is, and you have to remember the name of the method, and there will be questions. Now questions will be theoretical, some questions will be theoretical, most questions actually. After what is questions will be asked on anemia, types of anemia, classification of anemia according to the clinical classification of anemia is according to the hemoglobin percentage. Like what is the normal hemoglobin count, what do we call as mild anemia, or if you are asked to classify you have to tell mild, moderate, severe. What percentage? 10 to 12 usually it is called mild, below 10, 8 to 10 it is moderate, and below 8 gram percent it is called severe anemia. Now you will face questions on anemia. What is the commonest type of anemia in phase T? We see get subjects in our country, subjects or patients in our country, anemic patients, commonest type, that is nutritional anemia due to iron deficiency. So there, due to iron deficiency, formation of hemoglobin is hampered, and what we, so hemoglobin percentage is less than what it should be. So iron deficiency anemia is the commonest anemia. So these types of questions you will face, and if you answer well there, if the examiner wants to give you more credit, then he may ask some tougher questions also. But first there will be some common questions, like these are the common questions. You first, on the experiment, on the practical experiment, what have you done, what is the name of the method, what acid did you add, what compound was formed. Already in the name of the method you use, acid hematin is the compound form. So can we do it by other methods? Is it possible by alkali? Yes, it is possible, but here we do it with acid. There are other methods. So some 2 or 3 methods, you will get the names in the books, like alkali hematin method is also there. So you have to memorize just 1 or 2 names like that. So these are the asked questions, especially if you use the book by, if you go through the book by Dr. C.L. Ghai, then you will find a lot of questions, which will, some questions are there. I think for the hematological experiments related theoretical questions, whatever you may face, this, what is iron deficiency anemia, what is the commonest type of anemia, all these things you will get a lot of questions in the book by Dr. C.L. Ghai. So first you read from any book which you are using, but I would recommend that after, when you read for the second time, then you just go through the book by Dr. C.L. Ghai on all these topics, these hematology topics. Also other topics, but for now the hematology topics. So for when you read for the second time, you go through that book, just collect it from the library, or if you have a PDF of the earlier edition, now it is the 10th edition is going on. If you have a PDF of the 8th or 9th edition, it will do. If you don't have, I'll send, I have the 8th edition one with me, I'll send it to the seer, you collect it and you read from there. Just you don't have to, buying every book is not possible and it is not needed. Just you take the PDF and you can read it on your, simply on your mobile also. So just go through the questions and answers from that book, and method you can follow your book. Method is written in that book also, but method you can follow whichever book you are following. So this is estimation of hemoglobin. So this will take 20-25 minutes. There is very less, nothing to wait, just 10-15 seconds of mixing, like DLC there is, or TLC waiting in the middle, that is not there. So maximum 20 minutes it will take. And another is blood grouping. It is, hemoglobin has also been discussed in the theoretical classes, so everything will be complete by now. Hemoglobin theoretical aspects and practical aspects, everything will be complete by today. Blood grouping, there are a lot of things to study theoretically, blood grouping and blood transfusion, but just for the hematology experiment part is very simple. Blood grouping, I think basic idea you already have. How do we classify blood groups? Actually strictly speaking, blood grouping is, say you speak about the ABO system, ABO, ABO system was, who discovered it, or who first described the ABO system? Someone called Landsteiner in the year 1900, and RH system was described much later, which the blood A positive or B positive. So this positive negative RH, this is based on the something called the RH antigen. This came much later. So obviously by the same person only, but this was much later in the 1940s or something. But in 1900 he described the ABO system. Now ABO system, strictly speaking, blood group is ABO together as a whole. Just understand, or just remember, blood grouping is ABO together. The ABO system is one type of blood grouping. There are some minor systems also, many minor systems, but they usually antigen antibody or antigen reactions do not occur with them, so we are not that concerned. But there are so many systems, and these systems are called blood groups. Now what is A positive? Say my blood group, we say usually, commonly we say my blood group is A positive, or his blood group is O positive, or her blood group is B negative. So we say blood group, but that is actually the blood type. If we say correctly in medical terms, then we should say blood group are the systems, groups are the, so ABO considered as a whole is the blood group, and blood group cannot be individualized. That means ABO cannot be together for some particular individual. The blood group which we commonly say, blood group of this individual is A positive, that is actually blood type, not blood group. You just, again, I am speaking why that book is elaborate. If you go through the book by Dr. C.L. Ghai, just the practical textbook, not the theory book. Theory book is also there, but practical book by Ghai, these things are written. So we should tell what is my blood type. The actual ideal statement should be my blood type is A positive, or her blood type is B negative, her blood type is O positive. This O positive, A negative, A positive, these are not blood groups. Blood groups speak of the, refer to the system, total ABO system. Most important ones which cause antigen antibody reaction in the body. If the antigen is there and antibodies are formed against this, then which are causing major reactions, which are maybe sometimes even may be fatal. So that may be fatal, the person may die. So causing that, those are called transfusion. If it is due to mismatched transfusion, that means a B positive blood type person infused with A positive blood type. So there will be transfusion reactions. These major transfusion reactions are not, I should not say dependent on, dependent is not the correct word. Major transfusion reactions are related to these blood groups, ABO blood group and RH blood group. And blood groups are the systems, not the individual characteristic. Individual characteristic is the blood type. So major transfusion reactions are due to these blood groups or blood group systems. So we consider them, but there are other minor blood group systems also, which usually, even if there is mismatched transfusion, or just the ABO groups are matching, but those are not matching between the two types of blood, the donor's blood and the recipient's blood. In case of blood transfusion, we use these two terms: donor, from which the blood is, from whom the blood is taken, they are not identified. Later they are not. After the blood is processed in the blood bank, obviously they are still they are not identified, but it was taken from some person. So he or she is the donor, but they are the donors, and the person who receives the blood is the recipient. So donors, recipient blood, ABO and RH matching is usually sufficient so that major transfusion reactions do not occur. Now maybe ABO and RH has matched, but some minor group has not matched, but due to minor systems matching is usually not required. So we take into consideration, practically into clinical consideration, ABO and RH matching. Now as you all know, these antigens, you must have known, this will be discussed, blood grouping and blood transfusion, there will be 1 or 2 separate theoretical classes, blood grouping and blood transfusion. If it is possible in 1 hour, then 1 class. Then ABO matching, blood type we write like this, A, B or O, oh sorry, 4, A, B or O, 4 are there. Blood types, we are speaking of blood types. In layman's term, we call this, my blood group is this, but actually that is blood type. So how many blood types based on the ABO system? AB and O. So this is on the presence of antigen. Now antigen in case of blood, this blood antigen, this classification, antigen in case of this blood grouping system, antigen, instead of antigen, better we use the term agglutinogen. And agglutinogen reacting with a agglutinin, antibody is called agglutinin. The process is called agglutination. The process is called agglutination. So if in the A blood type, we should say, in the A blood type, what is the name of the agglutinogen? It is same, the capital A. In the B, it is capital B. So there are 2 types of ABO agglutinogens, A and B, on the presence or absence of which the blood type will be determined. So if there is only A present, the blood type is A. If there is only B present, then the blood type is B. If there are both present, both are present, then the blood type is AB. If none of these are present, then blood type is O. Okay, and Landsteiner's law say, so okay, Landsteiner's law says, which was, Landsteiner's law was given by him in the year that 1900 only, not after long before the RH system was discovered. So Landsteiner's law says, if one particular agglutinogen, there are 2 statements, if one particular agglutinogen is present in one person, one individual, on the RBC of one individual, where the agglutinogens are present on the surface of RBCs. So one of the, if you have to elaborate the functions of RBCs, then one function you write just to make it up, just to make up the paper. It is not actually a function, it is a characteristic, but it houses agglutinogens, it houses blood group agglutinogens, ABO agglutinogens. So main function is obviously carriage of hemoglobin, carriage of oxygen, and one of the functions is housing agglutinogens. So agglutinogens are present on the surface of RBCs. Landsteiner said the first law is simple law, if one agglutinogen is present, corresponding agglutinin will not be present. What is the corresponding agglutinin? It is called alpha. What is the corresponding agglutinin for B? It is beta. So if A is present, then alpha will not be present. And the second statement says, if alpha is present, that means if one agglutinin is present, then the corresponding agglutinogen will not be present. Simply he told this. Now he did not tell what will be present. At that time he said, if A is present, alpha will not be present. We know, but we know if A is present, now we know when the agglutinins were individually described, if A is present, this is present, beta. If B is present, what agglutinin is present? Alpha. And whenever there is a transfusion reaction, what is there? What is a transfusion reaction? Transfusion reaction occurs between whom? The recipient's plasma. What is present in the plasma? Plasma contains the agglutinins. Okay. It is the recipients. The recipient is, say, receiving how much blood from the donor? 350 ml or something. It is diluted in the recipient, say 4 liters, almost 5 liters of blood. Okay. So the donor's agglutinins, which is contained in the donor's plasma, are diluted in so much amount of plasma in the, So if 5 liters of blood, what is the amount of plasma in the donors? These things actually is not a part of the practical experiment. These things will be taught in the theoretical classes. So say out of 5 liters, almost 3 to 3.5 liters is plasma, say 55%. If not 3.5, 3 liters, 60%, 2.8 liters is plasma. So in 2.8 liters of plasma of the recipient, donor's plasma is very diluted. So the agglutinins get very diluted. So what happens is the recipient's agglutinins attack, or it should not say, if we say attack, the donor's agglutinogens and the recipient's agglutinin reacts. So it is the donor's agglutinogens which is important. Okay. So it is the major transfusion reactions occur between donor's agglutinogen and recipient's agglutinin. So if A positive individual is transfused with B positive, then recipient is A positive. So the recipients, this beta will attack the donor's capital B agglutinogen. Reaction will occur between beta and B. Reaction should also between alpha and A, but it is usually does not occur in case of the A positive person being the recipient. It usually does not occur because the recipient's donor's plasma is highly diluted in the recipient's plasma. But the donor's agglutinogen and the recipient's agglutinin will cause the major transfusion reaction. Okay, these will be taught by the blood group, what are the hazards of mismatched blood transfusion. This is a theoretical aspect. So today, what we just for doing the experiment, what we need to know, A type contains A and beta, B type even not these things, only the agglutinogen. So A positive contain A type of agglutinogen, B positive, not positive, sorry. A contains A type of agglutinogen, B contains B type of agglutinogen, AB contains A and B both, O contains nothing. And when RH system was described, in RH, what we called RH positive, that is A positive. This positive is RH positive. RH is after the name of Resha. It was first isolated from a specific group of one type of monkeys called the rhesus monkeys, RHESUS. So RH positive, there the anti-agglutinogen is D, capital D. And remember, A has a corresponding agglutinogen beta, B has corresponding alpha, and whenever Landsteiner's law was described far back in 1900, almost 30-40 years before, 40 years before RH system was described, Landsteiner established that there is some corresponding agglutinin which will not be present. But capital D agglutinogen does not have any corresponding agglutinin. In negative persons, then we say a person is B negative, he or she will contain B agglutinogen, capital B, but there is nothing called delta instead of D, corresponding to D, no agglutinogen. He will contain B and alpha, but no D, or he will contain B and alpha and D, but there is nothing agglutinin corresponding to D. Now what we do in blood grouping in the practical to determine it practically, that is there are antisera. These are artificially made antisera. Antisera, these are called antisera, plural, singular is antiserum, plural is antisera. Anti-A antisera, say this one is, I think, anti-A antisera. So this is not the natural agglutinin, this is just antisera artificially made, which will react with agglutinogen A. So it is called anti-A. This is not the same as the alpha agglutinin which is naturally present. This is anti-A antisera made from plasma. It may have some chemical features similar to alpha, that natural agglutinin, but this is artificially prepared from serum of human beings. So this is anti-A antisera. This will react with agglutinogen A to agglutinate. This is in vivo. In vivo means outside the body. Outside the body, anti-A antisera will react with agglutinogen A. So whenever anti-A blood is applied to anti-A, if agglutinogen A is present in that blood, then there will be agglutination. This is anti-B antiserum. Anti-B antiserum. If blood is added to it, if the B agglutinogen is present, then in that blood, then it will react to from that to that agglutination can be seen, and it can be seen with the naked eye, well with the naked eye. Okay, it does not require the microscope, but it is better under the microscope, but it can be seen with the naked eye also, agglutination. This is anti-D antiserum. If capital D, that agglutinogen is present, then that agglutination will be formed. So if this is used and blood is added, which blood groups do you think there will be agglutination? B and AB also, because wherever B agglutinogen will be present, the agglutination will be there. In this case, A and AB. So when what we do is, on the slide we take, usually we can do it on a single slide. If we just take precaution, we just to draw two lines in between, we draw two lines in between and put one drop of antiserum A on one side of the, or in one part, antiserum B on one part, antiserum D on one part, and just simply pricking the finger, we add the blood. We just add the blood to that antiserum. So if this agglutination is seen in both antiserum A and antiserum B, then the blood group is AB. We should not say blood group, blood type is AB. Blood type is AB. If it occurs in antiserum D agglutination, the blood type is positive. That means AB positive. That means if agglutination occurs in all, then the blood type is AB positive. Agglutination occurs in with antiserum A and antiserum D, but not with antiserum B. Blood group, the blood type is A positive. If the antiserum does not occur with antiserum A but with the other two, it is B positive. If it occurs with none, it is O negative. If it occurs with only antiserum D, it is O positive, because it does not, that means it does not contain both of the, either of A or B, that it is O group and it has occurred with antiserum D, so it is O positive. Very simple. If you know the theory, it is very simple. But these are not the natural agglutinins. It has been prepared from the serum. Usually it is not done from human serum, it is done from horse's serum also, but human serum, if it is done, so it has been prepared from naturally serum of the individuals, but it is not the same, exactly same as the naturally occurring agglutinins. It is the just artificially prepared antiserum A, and according to the agglutination, whether it has occurred or not, we can say it is A, B or D. So, but for the beginners, it is done just to understand, just to first to note how what is agglutination. Agglutination, it will fall small clumps. After adding the blood, small clumps can be seen, and that can be well seen with the naked eye, better under the microscope, but also under the naked eye. So to identify those clumps first, for the beginners, how it is done? It is done 4 different slides are taken, which is actually not required. 4 different slides are taken. On one slide it is antiserum A, it is one slide it is antiserum B, one serum antiserum B, antiserum D, and to note where agglutination, how it looks like, to differentiate it, on one slide simply normal saline is taken. Obviously normal saline has no agglutinogen, so it will not form agglutination, okay, with blood. So we add blood to normal saline, and just as beginners we note whether there is anything. So just the blood will look like something, there will be no that clump like structure. So we identify this is not agglutination, and we differentiate it with the agglutination which has occurred maybe in all of the 3 or 2 of the 3 or 1 of the 3 slides. Okay, we compare it with that normal saline slide. That's why for the beginners we take 4 slides, which is not actually required. It is well visible. If we take 1 slide and make 3 divisions, it is usually visible. Another point which should be mentioned, it is written, the books with most of you, this book by Dr. Pramanik, which is written over here. We can do, like we can prick the finger and directly, if we have already taken the antisera on the slide, we can do it directly from the finger. But sometimes without this, directly taking blood from the finger and putting it into the antisera forms there is chance of another process called rouleau formation, which is different from agglutination. So rouleau formation, agglutination, coagulation, all these are isolated separate terms. From the exam point of view, one common mistake is during this blood grouping experiment, instead of agglutination, you know what you are doing, but you say the term coagulation. Coagulation is a completely different thing. Blood coagulation is clotting of blood. The BTCT experiment which you did on Monday, clotting of blood, that is, clotting is coagulation. This is not coagulation, this is agglutination. And rouleau formation is vertical stacking of RBCs, which should be prevented during total count, so that we can count the RBCs conveniently. So for that, in Hems fluid there is sodium sulfate, that is rouleau formation. Now directly taking finger during this blood grouping experiment, directly taking blood from the finger may sometimes form this rouleau. If rouleau formation occurs, it may be, especially for the beginners, to differentiate it from agglutination. So usually blood is diluted in normal saline. Blood is diluted. Normal saline does not hamper the agglutinogen property, this agglutination property of blood. Just blood is diluted with some normal saline, kept for some time, and then the blood is added. But even if we directly from the finger, the agglutination is well visible if we do not confuse it with rouleau formation. But directly taking blood may form this rouleau and confuse us whether this is rouleau formation or agglutination. So we should not do. It is written in this book. Also, I think it is written in other books, but it can be done. But for preventing rouleau formation, we dilute blood in normal saline, keep it for say 5-10 minutes, and then rouleau formation is prevented in that way, and we can then add the blood to the antisera. So this is a very simple experiments, both hemoglobin and blood group. Today it's blood grouping. This is 11:54 in 1 hour. It should be sufficient. And please make it sure you do this, because I don't know, you may know that whether you can be present on Monday or not. On Monday again blood grouping will be done, but if you know that you won't be present, but don't take it as I would recommend. If you do today, then don't come on Monday by yourself. Only if you have the community medicine field work, you don't come, but again you practice it on Monday, and you may practice other things also. But by Wednesday, for you Monday, for your batch, Monday is the last class of hematology practical, for at least for the time being. Some topics are left, but at least for the time being, we will have to continue with clinical physiology classes. So if you do both today and you are present on Monday also, then there will be just revision of blood grouping, or maybe if revision of differential count and those also. The HOD sir will be present on Monday, but today you do both. 1 hour is sufficient, okay, and on Monday you revise. Those of who will not have that community medicine class. Okay, pay attention, everybody, attention. Stop, be silent and pay attention. What I forgot to tell there, those who have already done know, the strength of the acid taken is N/10. Okay, that is the standard. So questions may be asked, what happens if that means more concentrated acid, say N/5 taken or N/2, will the experiment be possible? The answer is no. So this is standardized with N/10 HCl, the acid hematin reaction. But another question is asked is, after acid hematin is formed, what we add is distilled water. Instead of adding distilled water, if we add more HCl, will the reaction with this experiment be possible or not? So what do you think is the answer? Possible or not? So it is written in the book, it is possible. So after once acid hematin has been formed, whatever we add does not, that means either distilled water or the acid itself does not alter the experiment. But why do not we use N/10 HCl? Why do we add distilled water instead of adding this acid only? Because acid is costly, that is the answer. Okay, so that is from the, to make the experiment cost effective, we add distilled water. But the strength of the acid is N/10 HCl, remember that. It should not be greater strength or lesser strength. It has been standardized by taking N/10 HCl.